very excited to join you all today. Thank you for the invitation and the ability to present in the seminar series. So today I'm going to give an overview of how I've been using neutron scattering techniques to understand what happens to wood as it absorbs water and as it's modified by treatments meant to impart decay resistance and make it all overall better performing. So as just mentioned, I am a materials research engineer over at the Forest Products Laboratory, which is a U.S. government lab that specializes in wood research. It's been around for 100+ years, still government funded. And I'm one of about 40 scientists, all working in different areas of wood research, economics, life cycle assessments, nanotechnology, composites, you name it. So when I first joined the lab, I joined as a grad student. And my research was actually focused on wood-water interactions. Most of it, I was really interested in the nanoscale component of it, and that's how I actually got started using neutrons. So I looked at using small neutron scattering to understand microfibril swelling inside wood cell walls, as well as X-ray diffraction to look at crystallite swelling, and also looked at the dynamics using neutron spectroscopy techniques like quasielastic neutron scattering. Today, my research is a little bit broader. I'm working on essentially the connections between nanoscale and macroscale performance of a couple of different forest products. So I'm using light scattering to look at cellulose nanomaterials research. In particular, I'm interested in understanding how we can align cellulose materials and form better tactoids and control their domain sizes so that we can use that for barrier coating applications. So I'm co-leading a project in that area with Carl Hauptmann and Ron here at the lab. I'm also working on using light scattering and now probably also starting to look at X-ray scattering as well, of proteins, so that we can make better soy protein adhesives that are more moisture durable. And so that's another area in which I'm working with Chris Hunt. And of course, a big area that's dear to my heart, kind of how it started, is using X-rays and neutrons to make wood products less susceptible to damage and understand how we can fine tune the things that the tree has been designed to do very well. And some of the specific areas that I've been dealing in lately in terms of the macroscale performance is, of course, it's ability to withstand fire and remain structurally sound, as well as biodegradation, particularly how it behaves after it's been exposed to different wood decay organisms, and of course adhesive bond-line failures. So why are we interested in this? Wood is a very interesting complex material that has many levels of structures, and here I have an schematic showing the different levels of structure for wood, ranging from it being an isotropic with 3 primary planes, and also having a cellular material with 2 types of cells. For softwoods like the ones shown here, the trachea is the most common one, and then when we look at those tracheates, we also have a material that is made out of a layered structure, and each one of those layers is made out of a nanofiber reinforced material with microfibrils that are made out of cellulose that have water accessible regions in between them that can absorb water. And this area in particular is where I was very interested in understanding our knowledge gaps, partly because wood decay organisms and any of the degradation elements will target these regions, and so will protection treatments that are meant to impart resistance and improve the performance of the products. But how they actually modify this nanoscale area was not as well understood, so I started really deep diving into this concept. And of course, as if it wasn't complicated enough, it's also hygroscopic. So of course, we need multi-scale tools so that we can really understand what happens to wood structure as it is subject to all this different stimuli. So different techniques are needed, and I started gravitating towards scattering-based techniques because of a few key advantages. One was that when I started looking at water wood-water interactions, I was quite interested in the idea of in situ humidity control. So being able to use scattering-based techniques means that you can study your samples in real-time conditions and in realistic conditions. Also, there's typically minimal sample preparation required compared to other microscopic techniques where you might need some heavy staining or surface preparation that may impair your ability to answer some of the questions you might be interested in. Of course, we are looking at using neutrons and X-rays because they interact differently with the material, and neutrons in general have higher contrast for wood, especially if we can use deuteration in our experiments. So now I'm going to talk about like 3 different examples of how I'm using neutrons in different cases to better understand specific wood properties. So my first, and the order of these is somewhat related to how difficult it is to maybe prepare the samples. So first, I started looking at thermonutrient imaging to study shard wood. The question that we're interested in is, can we measure the density change that probably occurs across the pyrolysis zone when wood has been exposed to fire? And the reason why we're interested in addressing this question, and this is a research project that's ongoing with my colleague here, Laura Hasper, as well as Nate Bickley. So we were interested in getting this information because this can be used to in current engineering practices and codes and to better understand when we should say like this is not going to be structurally sound. How are we defining when char is being formed? Since char is an insulating layer, it protects wood, but it's also crumbly. So to address this question, we were able to start looking at using neutron imaging. And by looking at the attenuation profiles of our samples, we were able to backtrack a density calculation compared to the original composition of wood when it's just virgin. And the ability of using neutrons in particular was that we have an enhanced contrast because we have a hydrogen-rich material, and we can start detecting more gradual changes in the density across the increase in temperature. So how did we do these experiments? To actually pick out those differences in attenuation, all the samples were the exact same thickness. So that meant that we had bigger blocks that were treated with a cone calorimeter, so we can subject them to a heat flux to cause the thermal gradient across the sample. And we also control the orientation during the experiment. So in this case, we were imaging samples in the transverse direction. That meant that the heat flux was coming from the longitudinal axis, but when we imaged the samples, we had samples that were about 10 millimeter thick. We used a bandsaw to actually cut samples that were like that thick and still whole, and then image them at dry conditions as well as after they had been wet, soaking them in 24 hours in water. And then we just wrapped them in aluminum foil after they had been like powder dry to not have like any excess water in the measurements, and went ahead and like measure them at a nearby facility. So, and these and use net thermal neutrons to do that. So it's a single exposure where we have like films in the back, and that way we can like retrieve the images. One thing that we wanted to see was if we could enhance the contrast by using drier wet conditions. And here's an example, and you can see like the differences in terms of contrast are striking. Wetting the samples definitely helped in terms of attenuation. Your measurements are more clear in terms of the image, but that also meant that voids, imperfections were much more clear as well, which actually made a little bit more difficult to do the quantitative analysis of like as you're going through different like late wood and early wood regions and along the different rings, which is how we like can then get this linear profiles. So for a lot of the subsequent experiments, then we really focused on the dry specimens instead, so that the effects of cracks was not as problematic in the quantitative studies. But this is like an ongoing area, and we're trying to start looking at how thin can we measure the samples. And because it's an easy experiment and to do, and it's fairly close by in terms like the facility. So it's right now is thermal neutrons that we can obtain via compact generator close to Madison, Wisconsin. we have started to include these experiments as some of those that we can do as part of our regular sample characterization techniques before we go to do X-ray scattering experiments, for instance, where we're doing scattering-based tomography to look at the differences in the crystalline domains as we shard the samples. We've also been using small angle neutron scattering to study changes in nanostructure. And I started this experiments first as part of my PhD, trying to better understand nanoscale wood-water interactions. That meant really controlling moisture-induced swelling and tracking it as a function of like time, humidity conditions, and a variety of different sample preparation methods. And we've also kept developing this tool to look at the effects of degradation as well as the effects of chemical infiltration. So typically in this scattering experiments, we have a neutron beam that's focused on our sample, and we just measure the scattered neutrons after they interact with our sample. We control the contrast in these experiments by using D2O, because that is how we get a better coherent signal. And there's two ways that we've done that. Typically, we either condition our samples inside a humidity control chamber, and I'm gonna go over in detail how our journey of designing sample chambers and our lessons learned. And then, or we can also do a more simpler experiment where we are just looking at the effects of a sample that's fully saturated. So our contrast then is just having this sample inside a titanium cell, for instance, where it's fully soaked in D2O. If we have enough contrast, then because we have this essentially tracheates up and down, and typically the alignment of these samples inside the cell or inside the humidity cell is important. And I'll show some examples of what happens if you don't take that into account. We will get anisotropic scattering. And the degree of the anisotropy here will depend on the alignment inside your sample and the alignment of the microfibrils inside the tracheates here, which are helically embedded along the longitudinal axis. The packing of those elementary fibrils inside the microfibrils will give rise to this diffraction peak. And once we anisotropically reduce this data, we can follow that peak position and determine changes in the swelling of these microfibrils. So because we were interested in nanoscale moisture-induced swelling, we looked at controlling the humidity during our experiments. So we could track the moisture-induced swelling and observe the changes in real time as the water vapor was going into our cell walls. So we first started doing this experiments when I was a second year grad student. And in our first iterations of doing our sample and humidity control setups, we started by using saturated salt solutions to equilibrate the humidity. And that worked really well in terms of how quickly it equilibrated. It involved a lot of offline experiments to make sure that the samples were being equilibrated quickly enough and that we understood when we should be doing measurements, since the neutron scatter measurements would be fairly long in comparison. We wanted to measure things that were equilibrated. But we also encountered some issues, which was related to the sample changes. So since we needed to open the chamber to change our saturated salt solutions, that actually proved a little bit more time consuming than we anticipated. So based on that experience, we switched to controlling the humidity inside the chamber using a generator instead. So the generators typically deliver a mixed flow of dry and wet air to control the humidity. And in this case, the generator is filled with a reservoir that has D2O in it instead of H2O. And depending on the beamline where we were using it, it was either hooked, the dry air portion was connected to compressed air, like in-house air, or in some cases, it was connected to a dry nitrogen tank. So In our second iteration, you can see that our sample was like much larger. We had many more spaces. It's still made out of plexiglass, and we had a big, long quartz window across the samples. That worked really well. We did, and here you can see like the RH probe that we're using as part of the feedback loop system to control the humidity. So as soon as we arrive to each humidity condition of interest, we would like change to the next condition. What we learned from this case was that having a larger sample like that really took advantage of the translation stage was like critical for us, since the longest time we're spending, we're just equilibrating the samples. So that allowed us to equilibrate many more samples at once. So once we had reached equilibration by tracking the scattering one of the samples, we could go to our next samples and just measure all of them. So that worked really well. So we learned that having a really, really long piece of quartz was perhaps not the greatest, since despite the fact that it did not look like it touched anything, it cracked. So we moved then to a bit more sturdy design, went away from the plexiglass chambers and started having something that was easier to machine, all based in aluminum. You still have your humidity probe location right in the center. We always kept the space empty so we could do our macro measurements, and then we had 9 sample locations for our samples. We also switched to individual windows so that it would be easy to replace any window that would be that needed to be replaced due to being broken or anything like that. Between like our first and third iteration, we also switched the spot size between our measurements so we could like do shorter measurements in comparison. And we also made sure that we could use this chamber across different beam lines. So by looking at having extra stand-up bases so that it would be compatible between different beam lines over at the Oak Ridge National Lab, which is where all the experiments were done. Here's at biosands and here's at EQ sands. All the samples inside typically wood swells, and that means that it can curl and that it can buckle, it can move. So in all these cases, we had our sample sandwiched in between aluminum sheets so that we could hold them, that would be easy to handle, and then also would prevent buckling or any twisting due to the swelling of the samples during the experiment. Then we like tape them all flat in the back of like each of one of the chambers using a polyamide tape like Captain. In the majority of the cases, our attempts were always to keep the samples as thin as possible to have enough signal, which often meant about 0.5 millimeter in thickness. It also was because like a thinner sample would equilibrate faster in our moisture uptake conditions. And we could reduce the effects of having extra cells along the path length and essentially augment our ability to discern small changes because we didn't have as many like fully dispersed conditions. We increased the sample surface area. So in every case, we try to fill up as much as our spot size with the sample. So in this case, we have an opening of 19 millimeters and our spot size of the beam was going to be 14 millimeters. So there was enough space to make sure that all our, we had a large irradiated volume. Despite the fact that our thickness was not as much. And for some samples, we also, depending on the samples that we wanted, what the question we wanted to address was we stacked sections to increase the overall sample thickness. And that allows to measure different questions. So if we wanted to compare the effects of having partially intact cut cells versus having whole cells, since each one of the tracheids is about like 50 microns, we could do stack sections, 25 microns, for instance. But to still have enough signal, we would stack those together to increase the overall effective path length. We also placed sections like side by side, depending on where the sections were obtained to increase that overall like irradiated volume. So there were a couple of things that we needed to take into account. I mentioned that we measured humidity control and we control it using D2O vapor. So One of the things that we had to include in our list of protocols of things to do before beam time to make sure everything goes well was check for the presence of H2O vapor. The reason for this is that having any H2O inside the internal reservoir of the chamber could lead to having less contrast, and having less of a scattering signal, having increase in the flat background that's due to incoherent scattering. So one of the things that we started doing was that prior to beam time, I would always arrive, and I still do a couple days ahead. I make sure to equilibrate my samples offline using the same setup I'm gonna have in the moisture-induced scattering experiments, and have them all dry in dry conditions so that I can start also in the right side of the sorption isotherm, because wood also has hysteresis, and as moisture-induced swelling could potentially have hysteresis. We did not know. So that was another thing that we controlled during our experiments. So we looked at having this, if we have our samples in a single one of the locations. Once we were able to start collecting shorter timescales, we were able to look at the roll of time. So that we could start looking at how like our sample was increasing in intensity as a function of time. As water is going in, we have better contrast, and the swelling between the different elementary fibrils starts to increase. So thus that correlation peak starts to become easier to detect. So in this case, I think this was like a 25% humidity, so the contrast was not going to be as great, but it allowed us to look at that increase in time due to just having the absorption. We also tried to look at the effects of being in the desorption condition, but that proved more difficult to control. Part of the reason was that there could be potential for condensation if we went too high in the humidity, and up to 95% is what we found was the less likely to have any condensation issues. So we also, like for future studies, typically just concentrate in having adsorption studies. So whenever I do a moisture-induced scattering experiments, then I'm starting offline early, equilibrate all my samples in a dry condition after they've been like soaked in D2O to try to like minimize the effects of having easily substitutable sites. And then I'll place all those samples inside my chamber, dry them for 12 hours, and then I will start equilibrating them in my first condition. So by the time my beam time starts, I am ready to start my first measurement. So the other item that we start then looking at is like, how are we preparing the samples? That's another thing that's going to raise variability and will affect our anisotropic scattering and our ability to really measure microfibril swelling. So there's a couple of ways that we can prepare samples for neutron scattering. We have used microtomes, we have used a guillotine, and we've even explored the use of Wilo mill. For most samples, typically we need to soak them first before cutting, especially if we're going to use the microtome or the guillotine, so that we can make them easier to cut and easier to do to have a smoother surface afterwards. Typically, by the time I get the samples to cut them, they have already been cut by the carpenter or by Joseph sometimes with either a saw, bandsaw, or just the pick, kind of like in the chisel, to get me a sample that I can essentially work with on the guillotine, for instance. So microtome we have used typically for samples that are going to be on the thinner side. So some of our samples that were meant to be like partially cut, so they were made out of like 25 micron sections where you were prepared using the microtome. Samples that have been degraded are typically really hard to cut thin. So we have found that the guillotine works really well for that. And we just try to make sure that the sample size is typically less than 1 millimeter for samples that have degradation, and 0.7 millimeters seems to work really well and is really easy to cut as well, as long as you're in the longitudinal plane for cutting. And of course, we have the Wiley mill. It was one of the very first things that we looked at to see how scattering from that would be affected, so that we could understand the anisotropic nature and see how things would change. And I have an example of what happens when, depending on how we prepare the sample. So here I have a late wood sample that has a low microfibril angle in it that's scattering from it. You can see like really nice sharp peaks, very anisotropic scattering. And then here I have a Wiley milled wood sample. So you essentially looks like a dispersible material. So to make sure that none of this potentially dispersible material would be dispersed after being exposed to neutrons, and you don't get into any kind of list with radiological folks, we make sure to just put it inside like this aluminum mesh, which is what you see here, and pack it inside there really finely. And that worked really well. We were able to get scattering from those sections. Nothing was lost, and we also didn't really get background from the aluminum mesh. And you can see like the 1D scattering signals from there. The positions are about the same, but the anisotropy, of course, is lost due to the sample preparation method chosen, so we don't have as much information. So for all future studies, and we really focus on having like whole samples that were like really carefully prepared. We also looked at the effects of like studying early wood versus late wood, which would have thicker cell walls in the case of late wood. And as you can see, thicker late wood cell walls have higher scattering intensity. We have more material, so we have better contrast. So for future studies, and here I have the samples, you can also see that they're visibly different in color as well, their density. So for future studies, and knowing that the differences were not strong in terms of the swelling, but rather in terms of the scattering density, we focus on late wood specimens. Another thing that we started doing as part of our preparing for beam time characterization was check for the microfibril angle of our selected growth ring. So we typically pre-select growth rings before we start a study if we're trying to look at the effects of chemical effects or degradation. And what we do is that we use an X-ray source, like here I have a Bruker D8, and we do either a SAC experiment or we do a XRD experiment to look at the spread in the 200 diffraction angle, so that we have an idea of how tight is a microfibril angle inside the cell walls. And typically we try to do low microfibril angles for all the studies. The reason for it is that our features are sharper, so it is easier to do better range in terms of the humidity, moisture-induced swelling, and it's easier to also detect small changes due to what we've done. So after everything works well, we were able to follow that peak as a function of humidity, and we can see it's falling at the elementary fibril scale. We were able to look at primary plane dependence. The reason we were interested in that is because bulk wood swells most in the tangential direction, so we wanted to see if there was an effect on whether or not our sections were from different primary planes. And what you can see is that the anisotropic scattering is depending on your primary plane selection. That is easier to observe in the lowest Q, and as evidenced by the difference in the diamond-shaped scattering. This is probably coming from all the lamellar structures that we have in the cell wall. But at the highest Q, where we have this diffraction peak, the transverse section is also susceptible to misalignment. And we were quite careful. We did microscopy studies, used staining to make sure that we were making really nicely aligned samples, and we still were somewhat misaligned with respect to the beam. Hence, we ended up with a symmetric pattern, as you can see here. So for future studies, we've really just focused on longitudinal sections, which are easier to prepare at all times and not have asymmetry. But we were still able to see that the swelling was not primary plane dependent. So that's another reason why we just stuck into longitudinal sections. We also were interested in looking at whether partially cut cells could swell more. Part of the reason is that cells near a bond line and an adhesive bond line and a composite they would be cut, so they wouldn't be completely intact. So we wanted to see whether or not these would swell more and experience more strains. And to test this, what we did was that, and I have here the actual samples so you can see side by side how they looked. We had our 0.5 millimeter whole sections of plate wood from the tangential longitudinal, radial longitudinal sections, and we made thinner sections that were just 25 micron thick using the microtome, but still about 25 millimeters across, depending on if they were tangential or in the radial longitudinal, we're limited by the growth ring size, of course. And stacked all the samples to reach the 0.5 millimeter total thickness, so we would have about the same amount of material, and stack them side by side very, very carefully. And that allows us to measure the swelling of the samples, and it showed that most of the cases, the partially cut samples really just swell more at the highest humidity conditions. And for the radial longitudinal, they're consistently a little bit less constrained, probably because they don't have constraints from delivery cells anymore. So that made us think that we probably did remove some hoop stresses from the S1 and S3 layers, but those were only discernible at the highest locations. So we kept studying then thicker sections that were easier to manufacture. We started looking at the effects of degradation. So for instance, if we have a brown rod decay, what we see is that typically your nanostructure will be lost, but preparing the samples is quite a feat. So, and depending on how the sample has been degraded, you will have different subtle changes in the nanostructure, but in general, that elementary fibril spacing will start moving. So it will either be able to swell more if you put the samples inside a titanium cell filled with D2O, or your peak will be entirely lost because you no longer have this long range order. So preparing the samples is a little bit of an art. Typically, they have been obtained by using dimensional blocks that were at some point fungally exposed, or in the case of archaeological samples, they are little pieces that were obtained from a larger object that was found in the bottom of a lake or something like that. So what we do is that since we know that things are not the swelling is not primarily plane dependent, we go ahead and reveal a radial longitudinal surfaces from one of these blocks using the guillotine. Typically, we have soaked them a bit of time so that it's easier to just cut a small surface. And then we air dry those surfaces ahead of time to make sure there's no curling. Sometimes we have had to use glass slides and things like that to make sure that they don't curl. Though samples are a little bit thicker than 0.5 millimeter, so that's not often a concern. Here I have an example of an unmodified sample obtained from one of these blocks, as well as a sample that was unmodified but also experienced a lot of degradation. So control versus non, and you can see that a 0.5 millimeter thick section, I was actually able to make it, but it did not survive transport. And this had about a 67% mass loss. So it was too brittle to transport or handle. So typically, we just make larger thicknesses so that we can keep them whole, and it's easier then to handle them, put them in the cell. Which typically, when we assemble these cells, you need to twist a screw component to actually seal them shut. So you have to make sure that your alignment is top notch, and sometimes we'll have to use a needle to go in and make sure that the sample is carefully aligned and try not to scratch the quartz windows, which happens more often than not. And then we carefully align those samples inside the cell. Part of it is because we need to take the instrument geometry as well into account. Many of the instruments have now detector wings instead, so we can take data simultaneously, but that also means that you need to think about where your anisotropy is going to show and make sure that your sample is aligned correctly so that you're not missing information. So degradation can impart these changes, and we can measure them if we are careful, so that we can better understand how durability is being affected, how the moisture properties are being affected. So what about the chemical modifications? What did they do? So we've also studied that, and we found that as a function of chemically modifying the sample, that elementary fibril swelling would also change. And in this case, we had to also use the titanium cells. Part of the reason was that humidity control experiments during COVID times was not an easy thing to do. These sections were like removed from those radial longitudinal sections from the soil blocks. So we had to align carefully inside the cells and try to like make different pieces all like fit to make sure we had enough material as well. So again, it's sort of an art, but that allowed us to detect how different effective treatments were able to preserve the nanostructure from the decay and compare then the swelling with unmodified dry wood and wet wood, and notice that effective treatments typically just keep the cellulose microfibrils dry. Another way that you can control contrast is by using adhesive infiltration or by using a chemical. So you can use a deuterium labeled chemical, for instance, phenylformaldehyde, and that way we can have a new source of contrast that's not necessarily D2O, and that way you have actually contrast in a sample that would otherwise have none. So if you measure these samples in a dry condition, and this is a phenylformaldehyde infiltrated sample, you'll see that a dry control sample that has nothing in it will have no contrast. We just don't have a signal, but a sample has been infiltrated with phenylformaldehyde will actually have a signal that looks very much like a D2O control sample, meaning that the phenylformaldehyde can go inside some of those regions that are typically water accessible, and it's mimicking and templating that structure. Another way that you can then also gather this information is by using contrast variation. So you can match the cellulose scattering by soaking your samples inside a solution of 35, 65 D2OH2O. And in that case, you will have also no scattering at the high Q range. So you have successfully matched out the scattering from the cellulose microfibrils. But for a sample that has been infiltrated with the phenylformaldehyde that's deuterated, your scattering length density is now higher. So you can see again that you have templated that material. So that's another way we can we have controlled contrast. So last but not least, we have also been using quasielastic neutron scattering and to measure biodynamics. For these setup, we also spent some time creating a humidity control setup, and because data is time of flight, we were able to bin it and just start looking at how it changed as a function of time, measuring every 15 minutes, so that we could actually like determine when the system had entered equilibration, since the dynamics and the length scales might not be as compatible with when we were expecting equilibration to be reached. And by doing that, we were able to like just track the broadening of this quasielastic neutron peak to determine how the dynamics were changed. Fitted to what we found were two different Lorentzian peaks, which we attributed to slow and fast water, and able to determine the diffusion of the translational diffusion of water inside the cell wall. So typically samples are mounted inside the cell. These are like 0.3 millimeters thick latewood samples that are fastened using this like aluminum sheet. So you can see a little screws there. The way that we did this experiment, since this is like a rather large area, about 2 by 2 centimeters, we made samples that were all like 0.3 millimeters thick, stacked them side by side, all from the same growth ring, so we could cover that entire area, increase our size, increase our effective volume, and also increase our scattering intensity, and held it flat to make sure that no curling, no movement would happen. We had a humidity probe that was here to just monitor the conditions, but it wouldn't talk directly to the generator that was being used to control the humidity conditions inside. And we also moved away from designing a chamber that was specific for one technique. So in this case, we designed a chamber that was compatible with a couple of different spectroscopy techniques. So here you can see the same chamber being used for neutron spin echo experiments. And all we did was use the same body, but now we have new windows that, instead of being fully made of aluminum, like the ones that you can see were used for plants, have now quartz windows, since that was more compatible. And we also have to change the screws. one thing that I would mention is like because of the geometry of the instruments, we had to use an equilibration volume to monitor the conditions. And this was really important because that way we could make sure that everything was operated how we could, and we could troubleshoot quickly, despite the fact that I had like done this offline experiment like a week ahead. It is having an equilibration volume with a couple of secondary probes was a great idea. For these experiments, H2O was good rather than D2O. So we actually used a water first reservoir just filled with D2O. And the generator, which you can see here, was connected to a nitrogen tank. And here you can see the humidity cell that is going inside the stick and then just gets lowered down with a crane. So takeaways. I have shown you this behind the scenes of successful scattering experiments, the stuff that usually doesn't quite make it into the manuscript. And some of the lessons learned from this have been like always try to reduce sources of variability. For us, we did that by like as much as possible limiting to a single growth ring. I will mention that for a lot of the degradation studies, that's typically not possible. But if you can, that is a great way to increase your chances of detecting subtle changes. We have also focused a lot on late wood just because we have a higher scattering signal. And whenever possible, characterize ahead of time a lot so that you can minimize also sources of variability. We do that in terms of like looking at cellulose microfiber angle, where we're pre-selecting samples, and whenever possible, we also like do X-ray diffraction ahead of time. We looked at some of the macroscale properties of our samples to determine which ones are going to be more likely to have differences, so we can like plan ahead and really have a schedule of how we're going to use our beam time properly. Optimize your signal to noise and identify all sources of background. Try to minimize them. Think about your sample geometry. Can you optimize at all your scattering signal by looking at that? What is your source of contrast going to be? How is it affecting your question? And run offline experiments. I, of course, had to do it because I did a lot of complex humidity control experiments, and those are not the bread and butter of typical instruments. But I think this is a true statement for any sort of experiment so that you can make sure that you get good, useful data. So that, I want to thank you all for your attention, and I'm ready for questions.